real quick, we want to introduce ourselves and thank you for joining us this afternoon, this Thursday afternoon. My name is Kevin Ong. I'm an extension plant pathologist and part of this group, which is called Air Fun.
Sorry, are we introducing ourselves or what the group is called?
No, what the group is called.
We are the Green Industry Extension Team.
Thank you. Air Fun can say it a lot better than I can. But there are several of us that are here today, and I like to get each of us to introduce ourselves. The person who is responsible for getting all of us together to have fun is Dr. Mengmeng Gu. So Dr. Gu, take it away.
Well, thank you, Dr. Kevin Ong, for giving me so much credit. My name is Mengmeng Gu. I'm a professor extension specialist in the Department of Horticultural Sciences. I'm a liaison between Texas A&M system and Texas Nursery and Landscape Association. That's all about me.
All right, and then moving over to Air Fun, who not only is a great guy, but he also doubles as a tech guy. So Air Fun.
Thanks. I'm an extension program specialist out of the Overton Center, dealing mainly with greenhouse, nursery, and ornamental pests and integrated pest management.
And I do also want to point out that all the cool graphics that you're going to be seeing about this group this year was put together by Dr. Becky Bowling, Grubbs Bowling. We prefer to call her Dr. Awesome. So Dr. Awesome, introduce yourself.
All right. Hi, good afternoon. Happy New Year. I am Becky Bowling. I am now an urban water extension specialist up at Dallas. I was our turf specialist for 2 years before that. I still get to play with turf within the context of urban water conservation, along with all the other things that come with water in the urban environment.
And we still have some folks, we have at least one that's not with us today, but one that we do have with us today is Paul. Paul, you want to share about what you do and how you are part of this crazy group? Or I don't know how you put up with us.
Thanks, Kevin. I am Paul Winski. I am a county extension agent down here in Harris County, and my focus is on commercial horticulture.
Excellent. Hey, Airfun, did you have something that you want to go over with the folks with what's going on this year? And you are muted.
There we go. Yeah, absolutely. Just before we kind of get started here, I want to give everyone an update on some changes we've been making for 2021. As you know, we kind of jumped onto these webinars as the pandemic hit and we could not have face-to-face meetings. And now we've been able to take some little more time on how to revise and refine this. So we are now going to have kind of a quarterly program already kind of pre-published before that quarter starts. Again, thanks to Dr. Awesome for these amazing graphics. As was mentioned, we're planning to, our team name now is kind of Green Industry Extension Team, or GIET, G-I-E-T. And Chat with Green Aggies is going to be one of our outputs as a team. So that's our weekly webinars that you can join, but we also want to invite you to join our Green Industry Extension Team Facebook page that's going to potentially have some other future programming as well. So we have that going, and you'll be able to find these quarterly agendas on there, and you'll also be able to find more detailed agendas made monthly about those specific talks. And you'll notice that every single month we have specific themes for each week. So arboriculture, turfgrass, nursery, greenhouse, and landscape. So that's why you can kind of tune in to weeks that are specific to your part of the industry or your specific interest. We're also planning on providing CEUs, pesticide applicator CEUs occasionally. So keep an eye out for those opportunities. So for this month is January 21st, we'll be offering one IPM pesticide applicator CEU. Like I mentioned, we do have kind of a more cohesive social media thing going on. So this AgriLife GET, again, that GET standing for Green Industry Extension Team, is going to be consistent across all of our social media platforms. So you can find this Facebook page, which we invite you to please join. We also have an Instagram account, a Twitter account as well. as a YouTube channel that will be posting all of our future videos. So subscribe and add all of those. So here is the Twitter account as well. And lastly, the YouTube channel, which as you can see, currently has no videos on it, but this recording will be on there. All subsequent recordings will be on there as well. So that's all I had to present on that end. Back to you, Dr. Ong.
All right, I'm trying to pull up my slides to share with you guys. So I sort of volunteered to go first this year because I figured this is probably the simplest time for me to get that done. And I said, well, I could really cover and take a look at our data from the clinic and see what's been going on last year with tree problems. Are you seeing my video? There we go. I love it. I'm trying out new stuff and I'm learning new things too on the internet. So what you see here is wrong hand, a schedule of things that are going on in January. So today, I'm going to spend about 20 minutes or so, maybe 25. And if I get too long-winded, I know Dr. Gu will shut me down, but I do have to jump off at 1. But we'll take a look at the data that we have from the Plant Disease Diagnostic Lab. This is the diagnostic clinic at College Station, and look at all the tree stuffs and see what pops out. I think you might find that quite interesting. And hopefully I have one slide in the back, or at least we can discuss a little bit about how to deal with those things sort of in a preventative fashion, if it is even possible at all. And we always still want to keep the what we call a plant of the week type feature. And so Dr. Gu and Paul Winski will be talking about the Chinese Fringe plant and share about that. Hopefully what you also noticed from this schedule, and as Irfan has mentioned earlier, that we're trying to keep teams throughout the week. So this effort here is to direct our efforts primarily to the green industry. And so we want to make sure to keep some sort of, I guess you could say, routine that the first week of every month, we will touch on arboriculture type topics. Second week of the month, we should have a turf grass topic. And the third week of the month will be a nursery greenhouse topic. And the fourth week of the month will be a landscape topic. Now, I don't remember exactly what we're going to do on if we have a month that have 5 weeks, but I think on those months, we will still have a chitty chat. So do please, ask your questions, share your questions. If it's something that we can help answer, we would gladly insert it someplace if it's not the chitty chat program on that particular time of the year. All right, let's talk about clinic samples and top shade tree. And I put the word here shade tree because when we actually take data in, I use different categories to sort of try and parse out how we collect the data and then how we analyze the data in there. So I just want to quickly show you about the number of samples that we officially receive in the clinic in 2020. We got 2,664 samples. This is all types of samples. And if you look at the chart here on your y-axis, that's right, the different hosts, what I call host categories. What I want to point out is under trees, I actually have 3 different subcategories there. Palms, fruit and nut, and tree. Tree is just shade trees, mainly what you would call ornamental and landscape trees. And I shaded that in green because that is the grouping that we are going to take a closer look at. There were 591 submissions in 2020 from all around the state of Texas on issues dealing with shade trees. Now, I realize some of you will probably notice, wait a minute, under fruit and nut trees, you got over 1,000 samples. Yes, we did, but I need to let you guys know that about 700 of those samples were actually citrus trees. basically part of a survey conducted by the Texas Department of Agriculture for us to test for citrus canker and citrus greening. So the results from those actually had implications on quarantine type issues in the southeast upper Gulf Coast, southeast Texas area. Palms, we only got about 62 palm samples, and I'll share with you some of our findings there because I think it's pretty interesting to share with you what we saw in the plant clinic. So I call this what we saw in the plant clinic. So top 15 in terms of number of samples by tree type is listed on this chart right here. And if you notice, number 1 by 4, we got 142 samples of live oak, Texas live oak. Interestingly, in our database, we actually kept a Texas live oak and southern live oak. So and categorize them differently: Quercus fusiformis and Quercus virginiani. One thing that's interesting is we rely on the person submitting to provide that information on species. If there was none that was provided, we often assume that it is a Texas live oak. Hence, that's why you see a large number of samples in that category. Later on, as we look at the disease part, I just lump all the live oaks together. I figured it's probably easier that way to when you look at stuff in terms of what pops up, and there are patterns to let us see the trends in terms of what disease showing up when and where. Other things is you see that we also got a large number of red oaks. The oak, the fourth thing on that list is basically no species were given, so it was just dumped under the species. We did get about 30 samples of post oaks, and then we actually got a bunch of cedar elm. And most of the time with cedar elms, it usually comes in the later part of the summer where people are freaking out because the leaves start to look yellow and chlorotic, and oftentimes they will have some sort of leaf spot or some kind of rust on it. But beyond that, most of the time it is responding to just senescence. So keep that in mind. Those are the number of type of trees that we get in there in that previous chart, which I have colored in green. So out of that 500 plus, these are the top few type of trees that come in. Now let's break this down. And I realize in the title I put Texas live oak. It's not. It's that top 10 diagnosis on all live oak. So I just lump what we were categorizing as both Texas and southern live oak into this. And each sample will have actually more than one diagnosis. In fact, and so when you start spreading that out, there were a lot. When you look at the numbers here, what it tells you is what was the test of the diagnosis, and what was the number of diagnoses that we obtain out of it. You might notice and realize those numbers are not very large. Believe it or not, we got a number of those samples that come in basically had nothing wrong with them. It's probably just environmental stress or some other issues, but there was no pathogens to be detected. Now, I did put in two things in here because it pops out. Because one of the things that we do try to keep track of is the oak wilt testing. There are people that just send in tree samples and specifically ask for oak wilt testing and nothing else. And so what I wanted to point out was what was positive, what was negative, and where is my cheat sheet? Yes, we actually had almost 100 samples that were sent in for oak wilt. And this through all different kinds of oaks, not just the live oaks, but within the live oak, I believe we actually have something like, I think about 50 or 60 or so. No, 50 were submitted for oak wilt. And off that, 10 was positive and 15 of those were negative, but had symptoms that make us think maybe they should submit another sample. So negative, but leaves were showing suspicious symptoms. But by far, to point out here, that biggest piece of the pie is Anthracnose. 67 out of, I think, what is it, almost 200, a little over 200 of those samples of live oaks that were sent in um, had a diagnosis of Anthracnose. And one thing I do want to remind you guys, at the Plant Disease Diagnostic Lab, we do confirmatory testing. You're not going to get a diagnosis unless there is detection, observation, in other words, proof of that pathogen present. Now, I use the common name Anthracnose because that would actually lump together both the Discula quercusina as well as the Gliosporium. So you got a couple of different fungi causing a disease commonly known as Anthracnose pop in there. A couple of things that I do want to point out as well, if you look around this chart, there is, what is it, fungal twig canker and fungal, oh, I can't reach that. That gray part next to insect gall is fungal leaf spot, which is primarily Tubacia, or formerly known as Actinopelty. Not surprisingly, every year we would get a number of samples in, especially with live oaks, with insect galls, and typically at the later part of the season. So this is kind of sort of breakdown just on the live oaks. Let's see what else I got. Oh, now I did a little fun with the data, and I said, well, let's split that up and let's take a look at the most commonly diseases or diagnosis on oaks by species. So if you notice those two lines, the two longest lines, this one here is the live oak, and the next one, 2, 3 up, there is the red oak. And by far, when you look at all this, one thing to point out is the colors. That blue color, that blue bar is Anthracnose. Then the orange bar is Tubacia or Actinopelty. And then there is another blue or lighter blue is rust. Those are the 3 big things that we saw. And then occasionally in several of them, you'll see Phytophthora root rot too. It's kind of interesting to note that this past year, in all the samples that we got, so we get a lot of samples from Houston, Austin, Dallas, Fort Worth area. So you think about all the urban areas, and we're getting diagnosis of foliar type issues, and this actually started at I think the earliest sample that we got in for Anthracnose was actually in January, February, March, April, late April. So from the southern part of the state, and then the last sample we actually have recorded with Anthracnose was submitted in November of last year. So it's a wide range of when we get samples come in. Now, the thing that I think a lot of folks ask me is, can you use this to predict, to know when you see some of this stuff? To a degree, you do have to realize we are basing this information on what comes into the clinic. Some of this diagnosis, like Anthracnose or rust, is relatively simple to diagnose if the person know what they're looking at. And so we may not get all the samples in. There might be other issues that are easier to diagnose and perhaps more common, and we may never see that in the clinic. So all you get here is basically impressions of based on specimens that come into the clinic. Wanted to show you a few pictures here, and then talk a little bit about some of the other data that we got. So this is just a few pictures of the samples that came in. On top here, the picture on your far left is a combination of rust and Anthracnose on that same sample. And on the right, we get two different pictures of red oaks actually that basically was diagnosed with just anthracnose with discula. Now, other combinations that we often see, usually during the summer and later part, is anthracnose and powdery mildew. That's the picture on the left of the screen, the top one. And we also get anthracnose and tubacia. What I thought was surprising with the sample that is this picture right here, I had thought that we would also pick up Alcinoe quercusina, which is what they would call a target spot or what is it, little spot anthracnose, but it wasn't. It was just straight up discula that we could actually recover, and tubacia. So it's kind of interesting that what we were noticing is the symptomology, whether it's on live oak or whether it's on red oak. You get all the symptoms and you can make some guesses, but until you actually look at it under scope or trying to culture some of it, you can't really get to the exact culprit in terms of genus and species. The other thing that was kind of interesting is if you notice like this specimen right here, it really looked like symptom of scorching. So another thing that a lot of folks would ask us to test for is bacterial leaf scorch, something that's caused by Xylella fastidiosa. And I think what's interesting about that is we did have a few that came in and did test positive for bacterial leaf scorch, but that number was really small, and it's primarily where we get the positives is on the red oak. And I think a lot of times that when we get asked to do those type of tests, it's also related to the symptoms that is seen. And what I can tell you about red oaks and bacterial leaf scorches, oftentimes where you get a year that is really dry, this could be super wet in the springtime, but then it gets into the latter part of spring and through the summer where it stays hot and dry, and you start to see the scorching. Oftentimes, if that scorching shows up very early, we'll be able to detect Xylella from it. The question that I've been asked with that is, does it really kill the tree? My experience with Xylella on red oaks is I have not seen a tree die to bacterial leaf scorch. I've seen it look ugly, and in a year where they water it well, or if there's sufficient water and mild type temperatures, you don't even see the scorching. So who knows? Maybe it's just lurking in there until a year where there is some challenges with water. So take away from this, at least on the oaks. But before I go to this, I do want to point out one thing. One of the things on that list of trees was Cedar Elm. So we actually got 35 diagnoses on Cedar Elms, and believe it or not, last past year we get samples sent in when they looked at that yellowing and then scorching. We actually get quite a bit of scorching on Cedar Elm. And 12 of that 35 diagnoses that were made on the Cedar Elm was bacterial leaf scorch. Interesting. After that, we do get about 5 cases of Phytophthora type damage, at least recovery from the vascular system or the root system. And 3 of those were anthracnose on mostly Gliosporium on the Cedar Elm. Now, real quickly, I do want to share with you a little bit about palms. I do have a little bit of interest in palms in that I work with phytoplasmas. If you guys don't know what phytoplasmas is, it's what I would call a naked bacterium, a bacteria that does not have any cell walls. It's not culturable, so we have to detect it using molecular methods. In the state of Texas, we do have quarantine regulations regarding several diseases on palms. namely lethal bronzing and lethal yellowing. Both these diseases are caused by phytoplasma. Um, Basically, different phytoplasmas that they differ by a few base pairs, so they are genetically almost identical, but they are not. So there are two different diseases. Lethal yellowing is one that we have never detected in Texas. Lethal bronzing is a disease formerly known as Texas Phoenix palm decline or dead palm lethal decline that has been detected in Texas, and there are areas around Texas, such as in South Texas and parts of the Houston, greater Houston area, that's under quarantine for this particular disease. Now, the importance is this: in Louisiana, they have detected both lethal bronzing and lethal yellowing in the ornamental or landscape palms. So for Texas, we want to make sure that we don't ever pick up that lethal yellowing. We do know we have some levels of lethal bronzing. We are trying to keep it managed and isolate those spots where it has been detected. So last year at the clinic, we actually had 66 palm samples sent in. These are mostly from homeowners or consultants, um, of which the breakdown is in terms of lethal bronzing. Canary Island palm, so Phoenix cariniensis, 3 samples tested positive for lethal bronzing. Date palms, which is more common, Phoenix dactyliferia, we had 9 of those samples came back positive for lethal bronzing. And surprisingly, well, sable is another common host for this phytoplasma. Uh, we had 1 sample in, and it tested positive for lethal bronzing. And then we had 3 unnamed Phoenix species. I have suspicion that 2 of those were Sylvestris that came in at 3 tested for lethal bronzing. We did have 1 Pindo palm that tested positive for lethal bronzing. So on that part, we do know lethal bronzing is been found in Texas, is common, and some of those results that I just mentioned to you, part of the plant disease diagnostic lab, we do have to forward that result to Texas Department of Ag. So in some cases, it does expand the quarantine area uh, to encompass where the range of those phytoplasma are. At this point, again, let me reiterate, we have not found lethal yellowing at all in Texas, but we do know that we have pockets, places where we have detected lethal bronzing. Those are the 2 phytoplasma disease that has quarantine implications in the state of Texas. Um, that in mind, let's just, I want to turn our focus back onto what we see most in terms of the oaks. We got a lot of oak trees in Texas, and we got a lot of oak samples that were sent in our way. And the primary problem that we picked up last year were foliar problems, things that are happening to the leaves. And it was mostly showing up in the latter part of the season, so in the summer. So this would be your anthracnose, your tubercular, and surprisingly, rust. According to Dave Apple, Dr. Dave Apple, a tree pathologist, rust is not something that you see very often every year. And so last year was just, I guess you could say, environment parameters came aligned that we actually saw quite a bit of rust showing up, at least in the samples that come in. Some of the questions that have been asked to us as well as Dr. Apple was, could fungicide treatment have prevented this? This is sort of a complicated answer. If you do see it early and you spray it, would it protect the plant? Theoretically, yeah. Whether it's discula, or whether it's gliosporium, or whether it's tubercular, or whether it's cornichium, or Eurymyces, one of the rust, a broad spectrum triazole would work pretty well, provided you can get good coverage and good timing. We don't recommend those for the basic reason that in a type of urban situation, there's always great risk for drift damage or drift incidents. So as such, when you have an established tree in the ground, we don't recommend that. But which brings me up to the next question is, is this something you can prevent future occurrences? Um, well, the one thing I do want to point out before I give an answer is to basically consider what can be controlled and what cannot be controlled by you. So what I call control uncontrollable factors in disease development. Realize that there are certain things that are beyond your control. We have no way to dictate how much rain or how much water comes in the spring, or how it's being delivered. I think in many parts of Texas, what we do tend to get is all or nothing, where we get a whole boatload of it at a short period of time, and then nothing for a long period of time. Um, with established trees on the landscape maintenance side, some people might say, irrigation is sufficient to some degree. But if the tree is really huge and established, it may take more water than you can provide it at the given time. So those are things that you do not have control over. We don't have control over what temperature it is and how much the temperature drops or increases at any given time. But what we do have control is how we feed the plant and what we can put in the plant. So with homeowners, what's nice about it, and also with landscape maintenance folks, there are products that can be used as a drench in order to get certain types of preventative products in there. And primarily, a lot of this preventative products that are taken in systemically through the roots, I think you're going to be surprised. They're either going to be strobilurins or they're going to be triazoles. So you got your propiconazole, tebuconazole products, or your pyraclostrobin type, azoxystrobin type products. There, unfortunately, with trees, there's not a huge amount of study on pathogen efficacy or fungicide efficacy with certain pathogens because it's just slow. And on trees, realize that there's going to be an uptake. So it really depends on whether you're dealing with a very young tree or an older tree. But that is one option that is can be taken. Trouble is we don't have a heck of a lot of data to show whether it works or not. And realize too, even if you put this sort of treatment in there preventatively, but if you got all those environmental factors come in line and it just hit it just right, where it's so conducive for the pathogen to grow and cause disease, and at the same time resulting in some stress to the plant, making it more susceptible, you're going to have disease. But perhaps it might be at a lesser extent. Anyway, that's what I got to share with you guys today on what we saw about trees at the Plant Disease Diagnostic Lab in 2020. And so if you look at this slide, I do want to point out, thanks to Dr. Awesome and Dr. Gu, and also Airfund, we actually got a plan for the next 3 months. And the topics are kind of listed. So we're going to be taking turns taking approach to this. And this was put together for the next 3 months. So as we are going to sit down again, I think at some time in the future, to plan for the following 3 months, if there are topic areas that you think you would like to hear about, you would like to learn that might impact your industry, shoot it to us or put it on the Facebook website, because those are topic things that we can bring up at one of our Chat with Green Aggies sessions. So with that, thanks so much. And I think I'm going to unshare my screen and then let Paul start sharing information, or Dr. Gu to share information about the Chinese Fringe plants. And by the way, if you guys got questions about what we saw in the clinic, and so I'm going to have to jump up in a little bit, you can put those questions in the chat, and Airfund will make sure I get them, and we'll try and respond to you and I'll try and respond as best as I can to you. But I'm going to hang around for a few minutes. If I do jump off, Airfun tag you it. So Dr. Gu, Mr. Winski, you guys ready?
I think Paul is ready. I just want to reiterate, normally there are 4 Thursdays in a month, but then we have a month that we have 5 Thursdays, like April. April 2021 has 5 Thursdays. So we're going to use that extra Thursday, and that's where we have the Q&As. We will not have presentations. That's where we will, invite you all to come in to ask questions, and also we will answer the questions from the social media sites. So Paul, go ahead.
Okay, so under this new approach, since we're doing arboriculture, I got to pick a tree that I really like, I really enjoy, and that is the Chinese fringe tree, Cyananthus retusus. It is a deciduous tree. This was the one, this is our old office in Bear Creek. And this was always would put on an incredible show.
Hang on, Paul. I think you need to share your screen if it's not shared. Is it shared? Yeah, it is. Sorry.
It is. Okay.
Yep.
So this is the one that was out front of our office in Bear Creek. And you could see the flower production on that. It from a distance, it looked like a white cloud. These are usually produced in March, probably about the 2nd and 3rd week. It's got about a 2.5 week bloom period. Has a slight fragrance to it. Landscape height is about 20 to 35 feet. This was a really nice specimen. The other thing that I really liked about it is the bark is deeply fissured, so it's got a unique bark presentation. With all that pollen, when it is in bloom, it does attract pollinators. And the one thing I guess the other thing I really like is that this is one tough plant, because during Hurricane Harvey, this tree was completely submerged underwater for several weeks, and then it sat in water for probably several months, or at least a couple months. And last year in March, I drove by. We can't get into the area because it's closed, but from the gate, I took another picture, and that tree was in bloom just the way it is there. So I would definitely recommend this. I'm starting to see it a little bit more down here used in the landscape. There's a couple developments that I've seen it at, but it's a great producer. It has a great presence to itself, and as a specimen, it's one that I would definitely highly recommend. Mengmeng, you have any other comments on Cyananthus?
Yeah. So for this Chinese fringe tree, there is a cultivar called a Tokyo Tower, and that one is slightly different from this, the species itself. As you can see, the species is kind of like a little oval, horizontal oval shape, versus the Tokyo Tower is more vertical. It's more vertical, slender. So space-wise, that one is more. It gives you the vertical, growth habit, compared to the species itself. But just as the species itself, that it's very floriferous. Paul, in this particular plant in front of your office, the old office, does it flower in May?
No, it would be in March. We would see this one bloom in March.
So how does it compare to the native ones, the native Grancy graybeard?
Good question. I honestly don't know. I don't know. I am not familiar with that one and when it blooms, so I don't know. This was every March like clockwork, about the 2nd week of March through a good 2.5 week period. It was in bloom.
Okay, Brent. Dr. Brent Pemberton in Overton just put in the comment that Tokyo Tower needs a good period of cold for flowering, so might not flower as well in South Texas. That's a very good comment, Dr. Pemberton. So I actually do have a small, I say small, it really hasn't really grown that much. I planted a small Tokyo tower about 4 or 5 years ago. And at that time, I didn't have a good spot, and I just threw it at the corner. I just throw it at the corner, and unfortunately, that's a spot where it gets a lot of water in the winter time, and so unfortunately, that one that I had, it has not flowered. It has not flowered for 5 years, for 4 to 5 years. Uh, so and that could be due to two things. And one, it could be due to that, it's at a constantly drowning spot in the wintertime. And the second thing is it could be because College Station is the winter is not cold enough for it to flower. The person who gave me the Tokyo Tower Chinese Fringe tree is Dr. Dave Creech from Stephen F. Austin. He had such great experience of this flowering in Nacogdoches, Texas. So he gave it to me, but it has not flowered for me yet here. I may have to move it to a better spot, or it just may not flower for me. So that's my personal experience with Tokyo Tower. I don't have the straight Fringe tree, the species itself. And John commented that the deer really enjoyed this tree in College Station to eat. so John, let us know if yours has a flowered. What you may not have a Tokyo Tower. So, that's all I have.
Irfan, you're muted.
There we go. Thank you so much, Dr. Gu and Paul. Dr. Ong had to jump off, so I'm taking over here. Everyone, you'll notice in the chat that Dr. Becky Bowling has put today's survey in there. If you don't mind, please take the time to complete that survey. That helps a lot with our programming, to improve our programming, and also to demonstrate that what we're doing has impact or meaning, or provide feedback on how we can have more impact. And want to remind everyone that next week's session is on turf grass, and the specific topics are what's new in turf selection and what to plant where the sun don't shine. That's shade options. And again, thank you so much, Dr. Awesome, for that amazing little flyer. So there'll be these weekly flyers, monthly and quarterly. So this one right here is for next week's. Remember, every single Thursday at 12 p.m., you can tune in. We encourage all of our growers to join in via Zoom. You can also watch via our Facebook page, which is that Green Industry Extension Team, or through Chat with Green Aggies. And we also broadcast this live through Twitter as well. So you can kind of watch this from all over the place, including anywhere else that is shared from there. So I want to thank everyone for tuning in. Thank you all for the wonderful presentations, and we'll see you all next week.
Really
Except
quickly,
there is one more thing.
Yes. Well, Zach had a question. Mengmeng, I think, would probably be the best person to answer it. If you see it, Mengmeng. Hi, Zach, by the way. Zach is one of our extension program specialists. He works in our weed science program.
Zach, I actually don't know where's a good example. Where that actually a good example for that because I have is a bad example, in my backyard. So I think it was someone else. Was it John? John said it was a good one to eat in the College Station area, in the BCS area. But Paul, do you have the address of the the old office? Where that picture was taken?
Oh, he's muted. Or is he muted?
Is he trying to talk?
I think he was.
Oh, whoops. You should be able to unmute
I'm sorry,
yourself.
that was my fault. It was in off of Highway 6 in Bear Creek Park. And I forgot get the address since we've been out of that office for the past 3.5 years now, almost 4 years. But it was right by the community center. You can't get up. That parking lot is now gated off. You can't get up close to it. You know, you could see it from the road, definitely. And when that is in bloom, like I said, in March, you would be able to see it. I think it's 3033 Bear Creek Drive, maybe, if I remember correctly. And if not, if you want to, you know, send me an email or contact me, I can get you the address of where it is in the Houston area.
I think some of the comments about, you know, we haven't seen this a lot in our area, you know, in Texas or something. It probably it's an egg and it's a chicken and egg thing, and this is why you know like Paul, like our group is trying to promote some of these trees that, you know, they're really nice, and it will be a good plan. It will be a good candidate to diversify some of our landscapes. You know, personally, I love crepe myrtles, but you know, if I don't think that we need I think we just need to add more plants, you know, to the palette, and that's maybe something our you know, our nursery industry you know, could take on. That's all I have.
All right. Thank you so much for your question, Zach. And well, that is all then. I think unless there was anything else, if I miss, if there are any other questions, you can put them there in the chat. Otherwise, you can always leave a comment on that Facebook group or page, and we can perhaps address it at the next Chat with Green Aggies. So I want to thank everyone again for your time, and hopefully we'll see you again next week. Y'all have a nice day.